Okay, is everybody done with the quiz? We're good? Okay, today we're going to do some uploads because we didn't do any yesterday. Sorry. So before we start, we're going to start with some uploads. But first, do we have any questions from yesterday? We're going to clarify some stuff today, so that hopefully will help. So I think one of the questions on the quiz referred to, what is the ploidy of, or what does it mean to be 2n equals 4, right? So, just a minute. Yeah, much better. An organism can be referred to as, so we're just going to include right now the diploids. So we'll just talk about mammals like us, right? So you can say a giraffe diploid number is 72. Okay, I have no idea what a giraffe has, but I'm making that up. But that means it has a total of 72 chromosomes in all of its normal cells, and half of that, right, in its gametes. Okay, but you could call it. I could tell you that organism's haploid number is something, without me saying it's a haploid organism, right? Your haploid number is 23. That doesn't mean you're a haploid organism. So any organism can be referred to as their diploid number or their haploid number. If I say the organism is haploid, then it doesn't have a diploid number. Right? So just because I'm telling you, I don't even know, donkey, the donkey's haploid number is 31, doesn't mean it's a haploid organism. That just means its haploid number is 31. So for each set of its chromosomes, there's 31 of them, which automatically means its diploid number is 62. So anything can be referred to by its haploid number. That doesn't make it a haploid organism. So that confuses a lot of students. As soon as I'm telling you, that n equals 1 for this organism, that doesn't mean it's a haploid organism. I'd have to tell you something else about it, right? But you automatically know if I say n equals 1, 2n equals 2. All right, we'll play with that a little bit. So I'm telling you that some cell is 2n equals 6, and I want you to show me two cells. One of these in G1 and one of these in G2. As always, the unreplicated is the line, and the replicated is the X. So this is un, and this is rep. And then you're going to screenshot and upload this to today's Moodle thing, but the first one is actually one I moved from yesterday, which theoretically is going to work, but let's just check to make sure. I changed the... Yes. Oh, August 11th. Oh, no, August 1st. Okay. Yeah, so you should be able to post to the one that actually says Moodle upload 7/31/1, but it's under today's date. Yes. What exactly do you want us to do? So draw me what the chromosomes look like and how many there are in G1 of the cell cycle of a 2n equals 6 organism, and a cell in G2, how many chromosomes, what do they look like? Sure, sorry. Oh, yeah. And just the number of lines is good too. You don't, yeah, that's fine. But that's good. Then that helps you in your notes if you go back and look at it. Like, what are all these sticks? What the hell were we talking about? So sticks are X's, and right, we do know that that stick really is a double-stranded molecule, and it's really spaghetti shaped. But don't draw me spaghetti. You can bring me spaghetti, but don't draw it for me. So this is 6 chromosomes, right? And so then the S phase is between here. So we replicate each one.
Each one?
Yeah, everything, like, right, all 46 of ours have to replicate, and then we pull them apart, blah, blah, blah.
So there would be double the 6.
So there would be 6 of these.
Okay.
Right, because each one gets a sister, gets a partner. Right? Yeah. Sure. Yeah. Yeah. I just added them because I'm thinking we might do lots today to make up for yesterday. Yeah.
I'm still yet to learn how to use an iPad. Is it okay if I draw it and upload it?
Yeah, that's fine.
You don't care?
No, I don't care. I know, I had somebody draw it on paper. They forgot they didn't have their iPad, but they had their phone. I'm like, well, just draw it on paper, take a picture, and, yeah, it doesn't matter. It's just if it's in your notes, then it's all together. You don't have to go back and forth between. But you can do whatever you want. Oh my God, look at all these hands. No. Do 31st, number 1. Yeah. But it's on today's day. I just moved everything from yesterday to today so that we have 6 options to upload. We're just going to have an upload fast. It's okay. If you put it in any of the ones today, it's fine. It's best to go in order, then I can keep track, but whatever. Well, yeah, just making my life harder, people. Yeah. Yeah, something on August 1st. There's, I think, 3 that say actually 7/31 and 3 that say 8. Well, any of those. Just go from top down. Make it easier. Okay, so how many sticks are in G1? Yeah, so you could put them all together. You could put them all in different spots. It doesn't matter, right? Because really at this point, they're not lined up. They're not doing anything. They're really spaghetti all over the place, but we're drawing them so that we can count them, right? And so then what happens between G1 and G2? Yeah, S phase replication. So each one of those linear double-stranded molecules gets replicated. So now we have 2 linear double-stranded molecules that are stuck together. And I'm lazy and I draw it like an X, but we really know, like, chromosome number 1 actually got a partner, right, replicated, right? So this side is 1 and this side is the other, right? Just like if I was making the X, it's really, this is one of the chromosomes and this is the replicated, right? But it's just easier to do an X. So I had problems with sneaky people on exams, and I have children, and I've been around the block, so don't try to pull this crap on me when they don't know what they're doing, and they do this. Because the book kind of shows them like this, so it isn't really fair, but they go like this. And they're kind of close together, and they're kind of curved. Is that an X, or is that unreplicated? Is that 2 chromosomes unreplicated or 1 replicated? Because they're not really sure when I'm asking what, so then maybe she'll just give me the benefit of the doubt. Whichever one's right, she'll assume. No! That is just crap, and you get nothing. Right? It's got to be a stick or an X. And yeah. But then, and then there's like a little dot in the middle. Really faint. Right? Don't play stupid games like that. You either know it or you don't, and you take responsibility for yourself if you don't. And it's okay, right? You didn't know it, whatever. Again, that's rule number 2, being a jackass. Don't play that kind of crap. Right? That's like a write-in when I'm looking for something like transcription or translation and they spell it transcrillation. No. You know, we're not marking off for spelling, but it has to be clear whether it's transcription or translation. Right? You can't like fake something in between trying to get points. Right? Okay. Sorry. Blah, blah, blah. Okay. So this one has 6 X's, wherever you want to put them. Right? They don't have to be lined up. They don't, you know. Oh my God, I owe Dr. Stein $1. My phone's ringing. No, it's Dr. Stein. It's my husband. He can't keep track of my schedule. It's the secret pocket down here. All right, sorry. Okay. All right, so are we good with that? Does that make sense, right? We have to replicate each one. And so then somebody, I can't remember who came and talked to me after class. Remember I was talking about chromatin and chromosomes? Well, there's another term as well. And that's chromatid. Annoying, right? And so, where we go? Okay. So if we talk about chromatin, right, that's the spaghetti. We know chromosome is like the sausage, which, you know, generally if we can see it, it's the 2 together. And so a chromatid is really one of those guys. Okay, either one of these we'd call a chromatid. The whole thing together is the chromosome. And this matters in the next step when we start talking more about mitosis, because we refer to these as sister chromatids, the replicated partners of one replicated chromosome. So this is a sister and this is a sister, right? But if we also had, for most organisms, there's more than one chromosome. So if we just drew another guy, oops, over here, that's a fat sister, right there. So these 2 to each other are sisters. But this one to this one are not sisters. They're just chromatids, but they're not sister chromatids. The sisters are the replicated guys, right? They're exactly the same because we built them by complementary base pairing during replication, which we're going to learn about next week. Yeah. So the chromatin is anytime it's just spaghetti shaped. So anytime before prophase. So it could be replicated also, right? In G2, you know, I draw them as X's, but they're really spaghetti. It's just 2 spaghettis stuck together. Chromatid and chromosome is during actual mitosis. Yeah. Right. But again, I'm going to try, there won't be a multiple choice question, you know, what's the difference between chromatin, chromatid, and chromosome? But those terms will come up in a question, right? Sister chromatids do something. And if you don't know what that is, then you can't answer the question, right? And that goes for meiosis also. Would talk about the sister chromatids and the non-sister, and you know. So we want to make sure we're clear. So these would be non-sisters if you're talking about chromosome 1 and the other chromosome 1. Right? Here's chromosome 1 with its sister. Here's your other chromosome 1 with its sister. Right? These guys are sisters, but those 2 guys aren't. Okay? And I'll keep talking about that so, you know, it'll make sense. I hope. Okay. Okay, I'm going to tell you cells or an organism, an organism's haploid number, oh, I can't make it too big, is 4. So there's an organism, its haploid number is 4. I want, oh, that's a small one. Again, I want you to show me the number and the shape of chromosomes in a cell in G1, a regular old somatic cell. The organism's haploid number is 4. Just like your haploid number is 23. But we don't want to draw that many sticks. And then we're going to screenshot and upload. Woohoo, more points. Remember, all these are mammals, right? Even though it's fake, because mammals can't have that too, but yeah, so just like us. A normal vertebrate. Right? This is its haploid number is 4. It's not a haploid organism. It's just its haploid number happens to be 4. Just like you guys are not a haploid organism, but you have a haploid number of 23.
So Thursday after we learn about meiosis, you can give us a diploid number and say, well, we need a gamete?
You betcha. Yeah, after meiosis, I'm going to start with a diploid number. You tell me, walk me through meiosis. What do you end up with, right? And same with mitosis, except it's just a lot easier. Yes, sir.
Is it okay if it's like oldest, or does that matter?
Oh, wait a minute. Maybe I didn't change the date on that one. Sorry, I thought I changed them all. That one says we're screwed. I said, no, upload for you. Okay. Oh, there we go. Oh, it just says it's late. Yeah, it doesn't matter. If it lets you upload, just do it. Because here, I'll change it just so you guys don't get the other one I changed, right? Okay, now you shouldn't get the message. You said that number 3 was okay? Yeah, number 3 is okay. Ow. Oh, you're right. Okay, I'll fix that one too, so then we don't get the error. Oops, it's not January. Okay. So if our haploid number is 4, then we automatically know what's our diploid number? Yeah. So then how many sticks do we have at G1? You guys are so good. And then how many X's at G2? Ta-da! I can't see. Oops, that looks like a Y. Sorry. How lazy I get. That make sense? Okay. Yay. Okay, so I could be a knucklehead and tell you guys the haploid number is 1, and it's a tetraploid organism. How many chromosomes in a regular somatic cell? Oh my God, you guys are awesome. Ta-da. We won't do that one. You can write it down, but you don't have to upload it. And so if you're given diploid, you should be able to tell me haploid. If you're given haploid, you should be able to tell me diploid. right? Those are easy. Tetraploid, same idea. 4n equals 4, n equals 1. Right. Okay. So, yeah.
What if diploid is like an odd number?
Ah, that's a good question. His question was, what if the diploid number, 2n, is an odd number? The 2n number can be odd. Well, let's just do one, shall we? We'll just upload it. Okay. So what if 2n equals 3? Right? So a normal body cell in G1, G2, how many chromosomes are in the cell in G1? Yeah. And then how many are in G2? At 3 X's, exactly. So yes, you can have an odd diploid number. It doesn't affect mitosis at all. Right? Mitosis completely works fine with an odd diploid number. What do you think gets totally messed up with an odd diploid number? Meiosis, right? Because then what would this guy's haploid number be? Yeah. Right? If we were just doing the math, we'd have to say 1.5. But during the process of meiosis, when we get there, you'll see you can't have 1.5. Right? We can't rip it, well, theoretically, we could rip a chromosome in half, but that's not what happens. Right? The gametes would end up either having 1 or 2, which is not possible. Okay? Yeah, you can upload this one, so that'll give us some more points. While I'm babbling on and on. Thank you. So generally, if the diploid number is odd, the organism cannot get all the way through meiosis because it stalls at the stage where it's trying to separate evenly. Because there's no even separation. Usually meiosis stalls, we don't end up making eggs or sperm. And so that organism is sterile, right? Cannot reproduce. It can exist, but it does not reproduce. Very rarely would a sperm or egg make it through meiosis, but the gametes aren't going to be normal because it doesn't have all the chromosomes, right? So even if it tries to fertilize, it generally won't result in live offspring. Yeah. Insects are weird. Insects, a lot of them can exist as a diploid or a haploid, and so if they're in the, usually it's under stressful conditions, and so they exist, they do parthenogenesis, where the gametes become an organism, so they have half the chromosomes, and then those guys can only do parthenogenesis, they can't do sexual, right? So that's under usually conditions where either population, usually extreme lack of food or water, where that's better than not reproducing at all. Like, I mean, I would assume it happened because not enough could find each other to do the nasty, do the insect nasty, right, for sexual reproduction because of those conditions. So yeah, insects are weird. That's why when I'm giving examples, I'm like, vertebrates, we're all normal. No vertebrate can exist as anything but diploid, right? So there is weirdo shit out there, but, you know, for these examples, we're going to stick to that. So a really good example of an odd diploid number is the mule. Mules, right? So mules, by definition, are sterile because they have an odd number of chromosomes. Who's the mule's parent? Yeah, donkey and a horse. I believe I think horse has 64 and donkey has 62 chromosomes. So when those gametes get together, they end up leaving you with an odd number, an odd diploid number. So almost all mules are sterile. Yeah. So then that would probably be, right, so I have to think of an example. If the F1 self on itself, that ends up with an odd number, then those guys are done. Right? You know, so whenever you get to the point of having an odd diploid number, that's when meiosis falls apart. Yeah. Okay. All right. So when we get to meiosis, it'll be more clear why odd is bad. But for mitosis, it doesn't matter. Yeah. Yeah. For meiosis, it matters. I mean, the organisms survive happy-go-lucky, but can't make eggs or sperm because mitosis is fine. Right. So if Right, so this guy, right, the next step after G2 is mitosis. We go through all the phases, we pull apart the chromosomes, right, and so each one of these X's, if we give us the little middle, you know, essentially in Anaphase, each of the X's, one side of the X goes one way, one side of the X goes the other, right, towards the poles. And what we end up with, right, is two cells that are 2n equals 3. So the odd number doesn't affect mitosis at all. Because all we're doing, we have a single chromosome, we replicate it, we pull each apart. Doesn't matter if there's 4, if there's 6, if there's 5, if there's 7, if there's 9, 49, 42, right? For mitosis, it doesn't matter. Meiosis, it matters. Okay. Okay. So if you exist as a haploid organism, like the grasshopper or some other weirdo insects that do this parthenogenesis where the egg just becomes the organism it develops, it has half the chromosome. Right, which means it only does have one gene for every single trait, right? Diploids have 2 genes for each trait. Remember our earlobes? 2 genes for earlobe shape. 1 allele from mom, 1 from dad. Could be the same, both attached. Could be the same, both detached. Could be heterozygous, 1 detached, 1 attached. Okay. And so usually simple organisms or weirdo insect things when crazy stuff happens. Okay? So we're essentially going to ignore the haploid organisms for the most part in this class. Okay? And so we're going to be talking about diploid, maybe throw in a little bit of tetraploid. Remember the guys that can have multiple sets, 4, 6, 8, only plants can handle that. No animals can handle that kind of polyploidy. Okay. What do we got? Okay. Now let's walk through this first, and then we'll do some more uploading. Okay, so we're going to go through the events in mitosis. And here we go. Here's our chromatid baloney. All right. Yeah. And again, they're showing us a cell that has unreplicated chromosomes. So what phase would this guy have to be in? Yeah, it has to be interphase as our big umbrella. And specifically G1, exactly. And again, they're showing it as a line, but we know it's spaghetti, but they're trying to give us the numbers, right? And so it's called a chromosome. Talking about the whole thing now, because it's just one. Really just a linear double-stranded DNA molecule. And then in order to get to this one, what is this guy if it's got replicated parts, sisters? Yeah, the arrow has to be S phase to get to this guy, which has to be in G2. Right, getting ready to go into mitosis. If they're actually condensed down, then they have to be prophase. But so we'd have to guess. If you were shown this picture, it's either a representation of the chromosomes and it's G2, or if if I say this is actually what they look like, they're condensed, we have to be in mitosis. We have to be in prophase, which we're going to get to in the next slide. Okay. And so again, the sister chromatids, right, they're pointing to them right there. The whole thing is considered a chromosome, just like this whole thing is considered a chromosome. And then the centromere is the guy, the pinchy part that holds them together. Yeah, and that's where the spindle fibers attach to pull them apart. There's the actual protein parts that the spindle fibers attach to are called the kinetochore. The kinetochore is part of the centromere. So I think our book goes into that a little bit of that kind of detail. We do more of that detail in cell bio than we do here, but just, yeah, it's proteinaceous. And it's really where the two sisters are held together. Okay. So we're cool with that, right? We've done that a billion times. You guys are done with that. Stop talking about that. Oh my God, what the hell? Okay. And then the overall mitosis, just like we just talked about, right? So in this case, we'd still say that has to be G1. And then in order to get here, if they're telling us these are condensed, then it has to be in M. So somewhere S and G2 happened. They're just not showing us cells of that, right? They've condensed down into what we could see as those sausage X-shaped-ish chromosomes. And then the whole point of mitosis is to pull the sisters apart, right? So that chromosome, if this guy that's circled here is chromosome number 1, one of the sisters goes here, one of the sisters goes here. For this chromosome, one sister goes here and one sister goes there. For this chromosome, one sister here, one here. I'm using all these colors. For this one here and one there. Because we want the correct chromosomes to go to each cell. We don't want two sisters to go to one cell and no sisters of another chromosome to go there, because then it's a mutant. And if that happens, the cell at the end before cytokinesis essentially checkpoints and says, okay, this is effed up and you need to kill yourself. And so the cells go through programmed cell death, or apoptosis, for the betterment of the organism, right? So you sacrifice yourself to save the rest of the organism, right? A cell can't function missing a chromosome, okay? It can't function with an extra chromosome. Organisms, most mammals, including us, need to have the correct 46 chromosomes in every cell for survival, right? There are several exceptions to that rule, which include having one extra chromosome of chromosome 21, chromosome 13, or chromosome 18. Okay? So if there's a mistake, we're going to talk about it after meiosis. A mistake in meiosis, sisters don't separate, homologous chromosomes get screwed up somehow. If it happens to be 21, chromosome 21 that got messed up, and a gamete has 2 of those, the other gamete has 1, when it comes together there's 3, that organism has 47 chromosomes total, 3 of 21, that's Down syndrome. So not developmentally normal, right? 3 13s is either Edwards or Patau syndrome. Can survive, but very developmentally delayed, and usually survives for several months to a year max, right? It's a disaster. Chromosome 18 as well. Can survive to birth, but is a disaster, right? Not functional. Down syndrome, pretty darn functional, right? There's different severities, but there's developmental delays in the nervous system, central nervous system, lots of organ development problems, heart, lungs, liver. So there's lots of problems. It's not, it's not normal because there's that extra chromosome. Yeah. No. There is no, okay, I lied. Yes, there's a couple. The sex chromosomes, we can tolerate having extras and missing them. For the autosomes, the 1 through 22, there's no existing surviving organism that is missing an autosome. You can't be missing chromosome 21. You can't only have one, doesn't survive. Right? So you can't be missing any of them. You can't have 45 chromosomes if the missing one is anything other than the sex chromosome. Right? And the only ones you can survive having extra are 21, 13, and 18. For the sex chromosomes, you can survive having too many and too few. So if you only have one X chromosome and nothing else, that's Turner syndrome. You look pretty normal, you act pretty normal. Phenotypically, unless you're a geneticist, you wouldn't know, right? Usually sterile is the only. No developmental delays, no, nothing else, but just the reproductive system doesn't form correctly because there's only one X. Yeah. There's no true hermaphrodites in the vertebrates. Hermaphrodites meaning both functional parts, right? That doesn't work, right? So if you are XY, but your Y chromosome, the gene on the Y chromosome, the SRY gene is not functional or mutated, you appear female phenotypically, but your parts don't work. Like you're sterile. Because again, you need two X's to get a full functioning female reproductive system. You have to have a functional SRY gene to have a full functioning male. So there are, at birth they look female, and a lot of times, now everybody gets genetically, they do more stuff, but way back when, you didn't know until puberty, and they don't go through puberty. When your daughter doesn't go through puberty, what the hell, and they're actually XY. They were a boy raised as a girl because they're neither, because they didn't know. Now they kind of look and check all the parts and make sure there's, there's ultrasound, but 50, 60, 70 years ago, they can't look for your uterus, right? Yeah.
So you're saying that all hermaphrodites are actually guys?
So there's really no such thing as a hermaphrodite, except in invertebrate weirdo things like worms. They actually have functional female system on one end, functional male on the other, and they don't need anybody else. They can, have a partner and have action on both ends, or they just do themselves. Screw you, people. I'm on my own. Yeah, but not in, upper vertebrate organisms, right? So, biologically, chromosomally, you can be male and appear female. Chromosomally, you can be female and appear male. That'd be estrogen insensitivity, right? Even girls, we have some testosterone. Testosterone's not on SRY. SRY controls the level of testosterone, jacks it up, right? So we have testosterone. If our estrogen doesn't work, right, things get swollen down there and look a little phallic. And so at birth, you would appear male, even though you're female, but you don't have a functional female system because you didn't have the estrogen to drive the formation of the full vagina, uterus, and all that stuff, ovaries. Right? Because in development, we start out as neutral, and the ovaries and the testes are the same thing, and as you get the hormones, if you're the boy, they descend and form as testicles. In females, they ascend and form as ovaries. So it's all about what hormones you're getting. So SRY is controlling testosterone and jamming the boys to get the, as my son would say, wiener. Again, scientific terms only used in here, wiener. And if you're a girl, right, the estrogen drives you to get the vajayjay. And the rest of that stuff. Okay, didn't know we were doing sex ed. So excited. I have no idea. Oh, we were talking about chromosomes, right? And so 3 X's, meta-female. You're like super female. So usually fertile, but more estrogen than necessary, so super female. Yeah, super female. So I don't know, look around, check those. But it's hard to tell now because everybody's got implants and all kinds of crap, so, you know. You can't go like, look at her boobies, baby, she's triple X. Right? Is it triple X or is it silicone? I don't know, feel them. And then boys can be XYY, so that's super male, right? And so generally they look like super male, and so super tall usually, right? Very super muscles. So check out the NBA. Okay, that guy is 7'9". His parents are not even 6 feet tall. I wonder if he has an extra chromosome. No. It's within the realm of possibility. The technology does not yet exist to manipulate giant chromosomes into really anything, even in mouse models and that sort of stuff. It's coming, but yeah, I mean, all of that, the messing with human embryos is strictly prohibited by law, and it's extremely expensive, so, you know, a evil rogue scientist would have to be loaded in order to, like, you know, have a lab in their basement doing genetic BS to humans, right? I mean, there's novels, I'm sure, that you guys should write one, right? Yeah. We could get, like, super NBA and win all the Olympics because we're, you know, generating XYY. They're, like, 9 feet tall. It certainly does. Okay. Yeah, so, you know, when you see super tall people, be suspicious. Like, oh, can I get a little blood sample? I'd like to check out your chromosomes. Yeah, right. So now we don't, but I mean, technically, so if you do IVF, right, where it's sperm and egg in culture, right, what normally happens is they're grown for several days until they're to at least the 8 egg cell stage, and then you can pluck one of those cells off. And so then you can look at the chromosomes to see what that embryo, what is that embryo, male, female. You can look, you can sequence the eye color genes to try to figure out what that embryo's eye color would be, right? Or anything else. You could look for all of the genetic disorders. You could look for anything that we know a gene exists for, Huntington's disease, sickle cell, any of that kind of stuff. You can't fix it. No, but you cannot use that embryo, right? So that's how you could, that costs so much money, you'd have to be like the millionaire, billionaire people to get a designer child. And then you'd have to find an unscrupulous, right? You'd also have to find an unscrupulous fertility doctor, which we know those exist because we hear all the stories about, right, that guy in Orange County 10 years ago or something who had 150 offspring. Because any woman that came in that needed a sperm donor, like it was the husband's problem that was not her, it was his sperm sucked and they couldn't do anything or whatever. Or a single mom just wants a baby. So instead of like, you know, you order which vial you want from the directory, yeah, and so he, you know, signed off, but he used his. Yeah, gross. Yeah, look it up. There's a picture of him sitting with like 50 children. So apparently it was, some of the moms figured it out because they, yeah, it didn't look, you know, they ordered a blonde, blue-eyed, you know, man's sperm and they got something, but it was, their kids looked similar to these other patients' kids, more than to them and what they thought they should be. And so they figured it out. He's in jail or something. I think he paid fines and whatever, but still. So bring your own sperm if you're going to do that. Get a private donor. Yeah. Well, I mean, yeah, I mean, they didn't all look exactly like, it was, but you know how you can see, like, in a group of cousins, they could all look very different, but you might notice they have the same kind of eye shape, or, or a mannerism or something, even if they look, so if you see enough, yeah. So, yeah, that's gross. Yeah. Yes, so there's all these crazy identical twin stories too, which we could totally get into, right? Where twins separated at birth end up marrying women that have the same name, have the same jobs, they both grew up and work the same jobs, like, right, what is an identical twin? A genetic clone. So things that we think might be nurture are hardwired. Right, are hardwired genetically. That person, except for any mistakes or, not mistakes, but changes during development, they're genetically identical to you. So you have to be careful if you have an identical twin and they're evil, right? Because their DNA left behind at a crime scene is your DNA. And if you don't know they exist, they could go like on a murdering spree and you're going to jail. Okay, I digress. Let's move on. I could do this all day, but we got to do something real, right? Okay. So let's do this. Since we just did this, let's do another upload. Woohoo! Okay. So let's do the overall, and let's go with something, let's do 2n = 4 just because it's easy. Right? And we're going to start with a cell in G1, a cell in G2, and then after cytokinesis. Okay, and again, you're going to show me. And so this kind of picture is extremely similar to things you might need to draw on an exam. Hint, hint. And again, I could, this time I'm trying to be nice, right? I give you 2n = 4. But I could say the haploid number of this organism is, right? I could have said, their haploid number's 2, show me a cell in G1, G blah, 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 and you'd still go, you'd have to start. Right? No, just, these two here are just after cytokinesis. Right, I'm skipping all the stupid phases in between. Right, so this is really M, mitosis. So I just want you to show me the end product after mitosis and cytokinesis. What do we end up with? Nope, you can make any old colors you want to. And then screenshot and upload this bad boy. That's a good question. So the other common misconceptions of this whole process is that the cells really exist in any of these stages, right? It's a continuous flow, and scientists have put a window on each step. So it's not, that isn't to say that the cell doesn't stall and relax and check, right? G1, make sure it's big enough to get through replication. If it's not, it'll stall there until it is, right? Replicate G2, there's all these checkpoints to make sure are all the chromosomes replicated correctly, blah, blah, blah. Okay, now we can start mitosis. But the process of mitosis, getting through prophase, prometaphase, metaphase, anaphase, telophase, and then finishing cytokinesis, is just a continuous process. We just put a line, okay, this part here is anaphase. So there's a range of where I'm not going to show you a picture where it's very close. Would you call that anaphase or is it telophase? You know, it's like a nanosecond beyond one and before, you know, like, because, and it depends on which book you read and what you look at. In telophase, most of the books still show them as a real chromosome, mostly because it's just easier to keep track of. But yeah, once they're sort of almost there, they start going spaghetti, chromatin. So it's definitely during telophase. By the time they finish cytokinesis, they're essentially all of a sudden, G1. You know, like, so again, yeah, that transition, these aren't like, ooh, I'm in prophase, I'm going to hang out with, you know, it's just, we've just marked them at major milestones of what's happening, just to help us categorize when does the condensation occur, prophase. You know, what's the next step, pro-metaphase, right? Pro-metaphase didn't exist when I took this 800 years ago. It was prophase, metaphase, anaphase. Now they added this pro-metaphase because it's something important happens there. They want to pull it away from metaphase. Metaphase is just line up in the middle, right? Prophase, pro-metaphase, we're attaching the spin. So, yeah, but it's really just a... So we're going to go with, for answers to questions like when does it actually disintegrate, we're going to go with whatever our book tells us. If it says disintegration starts in telophase, that's what we're going with, right? Because that would be the standard answer for any standardized test is, you know, even though we know it's this transitional process. Okay, so how many sticks do we have in G1? And then what do we got in G2? And then what do we got in these guys? Right. And so then 2n equals what down here? Yeah. So we started 2n equals 4, and we end 2n equals 4. It's exactly what we want to happen for mitosis. Right, let's write mitosis so we don't get confused later. So what's 2n equals what here in G2? 4. Oh my God, you guys are the best things ever. Our DNA content has doubled, right? But we still count 4. So if this, like, it's still a problem, think about counting the centromeres. That's the number of chromosomes there are. Okay, rather than thinking about chromatids as the number of chromosomes, think of the centromere, because really these guys have a little guy. We don't show it. It's kind of like their belly button. Right? So there's 4 centromeres in G1. There's 4 centromeres in G2. There's 4 centromeres after cytokinesis. And then that essentially means these guys are back in G1. Or G0. Right? Yeah. Okay. But we do have twice as much DNA because each chromosome now has 2 sisters. So when we get to 360, they play those games. DNA content versus number of chromosomes. I'm not going to play those games with you guys in here, but I want you to understand that so that the next level doesn't, you know, become a problem. And that there has to be twice as much DNA. We replicated chromosomes. Okay. Okay. All right. So interphase, right, blah, blah, blah. We know really is G1, S, G2. We've already talked about that, right? Here's our spaghetti-shaped crap. So it looks, in the real world, G1, S, and G2 look exactly the same under a microscope. You can't see any difference. It's just a whole bunch of crap. Each one really has the 2 sister chromatids. We can't see them. Here's something annoying. A centrosome. Really, can they come up with other names for things in this cell? Chromosome, centromere, centrosome. Honestly, you're killing us. Okay, centriole, right? Just stupid. So centrosome is made up of centrioles. Right, that's these guys. Okay. So So think of if, all these words are just blurring. Centromere, think of middle of the chromosome, right? So centromere with the M is in the middle of a chromosome. It's the chromosome's belly button or something like that. I don't know how to remember centrosomes and centrioles. They're not chromosomes and they're not centromeres. They're the other thing floating around. Okay? So if you're asked about what's important during interphase, these are the blurbs that a standardized exam would want you to know about interphase, right? In addition to G1 and G2 stuff, which they don't even talk about. Okay. And then actual mitosis starts with prophase. Right? That's when they start condensing down. Now they really, right, you can see, whoops, you can't see like that. If we draw the centromeres on them, right, you can see the sisters, they look like sausages. The mitotic spindle begins to form. So again, these are the important facts, the stupid memorizing stuff about prophase, okay, that we need to know. Right, these guys are started moving to the poles. The mitotic spindle fibers, those are the guys that are going to attach to the centromere, to the kinetochore, and literally bring the chromosomes to either end. So they have to get set up at the poles so they can bring the chromosomes apart. So here, right, the kinetochore is part of the centromere. So the centromere is the whole belly button thing. The kinetochore is where the spindle physically grabs the centromere, that part of it, the proteinaceous part of the centromere. Okay. So pro-metaphase they pulled out because there's lots of action going on. And they want you to remember this is where the nucleus dissolves, right? That double membrane around the DNA dissolves, goes away. It has to, right? You have to pull chromosomes to either end of the cell. If they're stuck inside an organelle, that's not going to happen, right? We want that to go away. And then this is where also the spindle fibers actually hook up onto the kinetochores. So each, to help us, I hope, and not confuse us, right, if I draw our giant close-up of our sausage, if we call this whole structure there the centromere, and then I'll color the kinetochore, so there's two, because the spindle fibers need to connect to one from one of the centrioles, centrosome thing, and then the other kinetochore needs to be attached to the spindle fibers coming from the other side. So that, right, if we have, that stupid thing and some fibers, and then, I'm bad at drawing that, and then some fibers, right, we want this one to be pulled to that side of the cell and this one to be pulled to that side of the cell. If that gets screwed up, if the spindle fibers from this side grab both of those, the whole chromosome is going to go to that side. That's called a nondisjunction. We didn't disjoin. It's like a double negative. Nondisjunction means they stay together, right? But the idea is to disjoin the two sisters. So this is the critical stage where it has to grab a kinetochore from one sister, kinetochore from the other, and pull them to the opposite poles. Right? If that gets, Stein's favorite word, effed up, and goes to one side, then we have too many on this side, not enough on this side. Neither cell is functional. Both have to kill themselves. Right? Both have to go through this programmed cell death, apoptosis, for the betterment of the organism, right? If you have too many chromosomes or not enough, you're a mutant. You're not doing what you're supposed to be doing. You could start going through the cell cycle and go crazy. Okay. So, right, spindle fibers from one side, one of them, other side, the other one. And then that helps them line up along the middle, right, during metaphase. So then that just keeps them nice and separate so they don't get stuck on each other and drag somebody the wrong way and all that crap. So now metaphase is just, they're in the middle. There's no other action going on there. Very lame. And then anaphase, right? So they're grabbed by their kinetochore, right? Those guys one way, these guys the other. They're migrating to the poles, right? So, they're usually shown about here. Again, are they telophase? Are they anaphase? Where is that exact transition? They qualify telophase when the envelope starts to reform around the nuclear envelope. Okay? And they show them as sticks here. The spindle, right, this guy's, all those fibers are disappearing. But they're still together as one cell. Right? This outer plasma membrane is still one, around both nuclei. That's telophase. And then cytokinesis is when we actually divide the cytoplasm. We actually have two functional cells at the end. With a full nuclear envelope, we're back to spaghetti, we'd be back to G1 or G0. And again, it's a transition, so in some books they draw telophase like in animal cells, the membrane kind of pinches. So, right, the starting of the pinching, the envelope is reforming, the DNA starts disintegrating. We'd still call this telophase because it's not completely separate. Once, you can really discern two separate cells, then we say it's done. And you can see here they're shown together, sitting next to each other, which is much more accurate than when I draw them far apart. Because in any tissue, they're packed together closely. They're all touching each other. Everybody's all friendly. Right? What are the only cells that aren't packed together that are free-floating? Yeah, are any blood cells. Red blood cells, white blood cells, platelets. Right? But when I draw it, I just want to make it very clear that they're totally separate. Cytokinesis has already happened. It's not like some weird, very, very end of telophase. Oh, I don't like it. Just to make it clear. But really, they're stuck together. So, in cytokinesis in animal cells, pinches in and makes a cleavage furrow, right? So, they call cytokinesis also cleavage, right? That's just what they would call cleavage happening. So, looks like butt cheeks to me. Okay. In plants, they don't pinch their membrane, right? Because we think about anything we know about plant cells, what do they have that animal cells don't have? Yeah, that solid cell wall, right? That carbohydrate-rich cell wall. The whole point of the cell wall is to provide structure. You can't pinch a cell wall. So what happens is they build a cell plate in the middle. They build new cell wall and new membrane down the middle. So they technically don't do cleavage. Cleavage is the pinching and the making of those two cells. Vesicles, Dr. Stein loves her vesicles. Okay. And so that membrane comes from the Golgi, right? Vesicles bud off, they layer themselves, pop open onto that new cell wall, create that new membrane on either side of that new cell wall. See, right there. I have two little videos of mitosis, which I can't seem to find. They're in my Dropbox, my CSUN box, my other computer, in the cloud. I don't know. I can't find them. So I will find them this weekend so that I can show you, and it goes through the process of mitosis. Very cool. And then this is just, fun little whatever. See? Ooh, sausages. So what is this? What is this crap right here? Yeah, and so this has got to be the stupid centrosome crap. Right, here's the other guy. Right, here's your sausages. What was this one? Oh, so here's interphase, right? Spaghetti, right? Blue dyes DNA. You can't see anything. So in this, is that G1, G2, S? Who knows? Okay. Oh my God, it's Metaphase. So you can see these bad boys are far apart, closer to the poles, and the green is actually usually dyeing actin, right? Some of the proteins that make up these fibers. And so you can kind of imagine, like here, right? You can see some of those fibers are attached to that guy, attaching to the kinetochore. And then metaphase, they're lined up, right? They don't look nice in a line like how we draw them, but you could see sort of the middle of each one, right? If you were going to mark the centromeres, they're all in the middle, right? And then their arms and legs are flapping around. They're sausage apart. And then anaphase are being pulled apart, right? So now the centromeres would be all on this side, and they're, kind of folded, kind of like the V shape in the stupid drawing. And then telophase, right? We see some pinching. Look at that. Right? This guy appears, it's still here, but it appears to be disintegrating like they claim, right? And the DNA looks hazy. You can't see distinct sausages anymore, but it's not spaghetti. It doesn't look exactly like interphase. Do we have another one for them? Yeah. And then cytokinesis, right? Two separate cells. We're back to this almost looking exactly like you can't see anything. It's a little like that they're still trying to finish disintegrating, but very close. And then this is my thing that won't play because it isn't freshly loaded. So I'll try to post them on Moodle and I'll show them on Monday in class. Okay, so Monday quiz. Yes, you'll probably have some flips, but we're amazingly enough not far behind yet. Shocking. Yes, I said yes because I know. Even with our whole hermaphrodite fun today, we still see. Oh, that's the good stuff, right? Yeah, and then read ahead for meiosis for a couple of easy meiosis questions on the quiz Monday. Right, not the nitty gritty, but what's the end product of meiosis, that kind of stuff. The phases of mitosis crap mostly on the quiz. All right, guys, have a great weekend. Oh, sure.
Out of curiosity, more than anything.
Sure. I love that.
What stage is crossing over happening between now and like the trait?
So crossing over happens in prophase 1 of meiosis. Yeah, so the